Hi everyone. In this video, we are going to discuss about the nucleophilic attack of the oxygen atom onto the phosphodiester bond, which is present in the incoming nucleotide that is added to the growing DNA strand during DNA replication. Okay, we know that ribose sugar, which has the OH group on its third carbon atom. So this hydroxyl group loses its proton. When it loses its proton, so that oxygen atom gets a negative charge, and when oxygen atom gets negative charge, we call this is a nucleophile, because this oxygen atom has electrons to donate to the uh, atoms which are need of these electrons. Those atoms are called electrophiles. Okay, so when DNTP is added to the growing DNA strand by DNA polymerase, this DNTP contains three phosphate groups. These phosphate groups are attached to the fifth carbon atom through ester bond. Okay, so these three phosphates are called alpha, beta, and gamma. So the phosphate which is close to the fifth carbon atom is called alpha, and the next phosphate groups are called as beta and gamma. Okay, so these three phosphates that are attached to fifth carbon atom in a DNTP. Okay, so this phosphorus atom binds with four oxygen atoms. Uh, so it forms covalent bonds with the four oxygen atoms. Okay, so these are all four oxygen atoms. They are in a covalent bond with the phosphorus. Okay, now that nucleophile, which is present on the third carbon, which is attached to the third carbon atom, it attacks on this particular alpha phosphate. That means it start forming bond with this phosphorus. So in a transition state, usually the phosphorus can form bonds with only four atoms, but in a transition state, in that state, the phosphorus can even form bonds with the five oxygen atoms. In transition state, this nucleophile forms a bond with the phosphorus. So immediately, this phosphorus can become, or can back to the its original condition, where it can binds or forms bonds with only four atoms. So during this, so this nucleophile forms bond, and the ester bond which is present between the alpha and beta breaks off, so that the two phosphates such as beta and gamma phosphates are released outside as a. The energy that is released during this nucleophilic reaction is used to form the bond between this phosphate and also the nucleophile oxygen as a ester bond. Okay, so apart from this, so the pyrophosphate further releases energy, because this energy is not enough to form the phosphodiester bond. So further, this pyrophosphate releases energy with the help of phosphatase enzyme. So that phosphatase enzyme breaks up the bond between the two phosphates, so that further releases the energy. Okay, so this is how the backbone of the DNA is formed between the sugars. So okay, there are no direct contact or chemical bonds between the bases of the same strand is present, but the bonds are the backbone that we call is present between the sugars.